the student density. So I think we have checked this, the web page, and we went, all of you have visited this individual kinds of cells. I hope you visited. You can raise your hands so that I will be able to understand.
Sir, I'm unable to download that or either view on this Chrome.
It happened with I think Atharva, but he was able to
Sir, I
do.
viewed it on Internet Explorer. That's the only way it opened on my PC.
Karthik, see, you try with Mozilla. Mozilla works. For me, Mozilla Firefox works. You can try with Internet Explorer too. Karthik, none of you, other than 3 students, uh, none has done this. None has visited this website. Okay.
Sir, it worked for me as well. I downloaded first Flash Player, then I downloaded Mozilla Firefox, and then it worked.
You can do the vice versa also. You have a Firefox and then you can download Flash Player.
Yeah.
So majorly these are Flash-based web tools. So you need a Flash Player. Some people may have an outdated Flash Player. They have to download the upgraded one. That's how you have to play around. Then you'll be able to see. Karthik, I hope you understand. Karthik Verma.
Yes, sir.
Would you try today?
Yes, sir. I'll try.
And let me know. Message me in this Teams app. Okay. So
Okay.
I believe all of you have done that, or if not, you will try to access this same portal again. And what I'm going to show you is just a recap of things. When you go to the prokaryotic cells here, you can see different portions. See, if you see this, this is a flagella. Okay, you do not have to zoom. So if you are curious about any portion of this drawing, you can hover the mouse over there. You do not have to click. If you click also, you will get some more information probably if it is available. In case of pili, there is no information available. So here is the overall bacterial cell. Majorly, we draw it in the shape of a capsule, and the blue color outline, when we click, it shows that it is a cell wall. And then when we click the red area, we call it a nucleoid. Can you see there is no specific difference or a dark spot, circular spot here in the center, as we have seen in the eukaryotic cells, right? So this is the nucleoid. It's not a nucleus, so what we call it is a nucleoid, nucleus-like material. Okay. Now there are a lot of dots inside the cytoplasm. If you hover your mouse, you find you can see it is ribosome. You can see that the lining is coming here. Ribosome, the sites of protein synthesis, right? And do you see any other organelles inside? When we say that these are the parts of a cell, bacterial cell, we normally call them organelles. Okay. For human, the parts of human beings we call organ, and in case of bacteria or individual cell, we call them organelles. And this spiral, Swiss candy shape structure is called a mesosome. So it's a very curious contraption. Many of the scientists, we don't know how does it work. Some role has been established, but not exact one. So sometimes it has got some role in transport, some role in. Many people believe it is there for oxygen. Many people believe that it gives a basic support while the DNA is or the nucleoid is replicating. Replicating in meaning it is copying itself into two nucleoids, and so the but you can see these contraptions in many vector. Many vector you don't see also. Okay, so this is a typical bacteria. All bacteria you may not see this pili, you may not see this flagella. This is a flagella which helps the bacteria to swim very fast. Okay, in water or in watery medium, it can swim very fast. As flagella. Often flagella are present all around its surface, or often there is a single central flagella. And then there are these pili. Pili are small, here you see protrusions of the membrane. The bacterial membrane itself protrudes out to give a appearance of a pili. So what are the functions it has? First, it helps in additions to multiple surfaces. And second, it also helps in sexual reproduction or sexual conjugation in bacteria. When we say sexual reproduction or sexual conjugations, we actually mean here is that there is some way one individual's nuclear material is being transported to another individual, or somehow nuclear material is being exchanged. Okay, so immediately you do not see new individuals forming, but nuclear materials are being exchanged. Okay, cell divisions or vegetative reproduction, we see the same nuclear material is being copied. Here, in case of sexual reproduction or sexual conjugation, here you see the DNA is not pure. It will be containing impurity, or we can say some sequences from other species. Okay, this wasn't discovered before last 50-60 years. All of a sudden people working, no, maybe 80 years. Yeah, all of a sudden people working in a very interesting story here. It is that one scientist, I'm forgetting his name, he was working with a disease causing bacteria. Now, when you say a species of bacteria, that particular species, all of its variety of the species, we call it individual. There are varieties of the same species also in bacteria. And so in microbiology, you cannot really distinguish so much that they are not mating, so they cannot be called species genus. So we try to match their RNA and all. That's how we determine their species. And each species have subtypes or divisions also. So one division or one kind of that species of bacteria can infect an individual. Another variety may not infect people. In our body also, we have, I just told you, there are 10 to the 12, 10 to the 13 or 14 varieties of 14 number of bacterial cells or microbial cells in our body, which is more than our eukaryotic cells present in our body. So how we are surviving? Because many of those bacterial strains aren't infecting us. Does the species are completely non-disease causing? No. These species, there are some strains which can cause disease, and but the one which were staying with us aren't. So all of a sudden some changes happens, and a disease causing similar species occur. Now here, same thing. There is this disease-causing strain of bacteria, a kind of bacteria, and there is non-disease causing kind of bacteria. Now what he did is he killed this, um, what do you say, disease-causing bacteria, and then he kept with the non-disease-causing bacteria. So when the killing actually meaning majority of the DNA or the cell will not be able to reproduce or not be able to divide by itself. Fine. So when both the kinds of bacteria are put together, what happened is that the non-disease causing bacteria start causing diseases. And they found that these pili were responsible, so that although the disease causing bacteria is unable to replicate, multiply its number, but silently it has transferred its virulence property or infective property through the sexual conjugation to the non-virulent, meaning non-disease causing strain. So it was a very interesting experiment, but with that we got conclusions that pili has multiple functions. So I'll just take you to the microbes once more, to the slides, so that I can give you a bit more details here. Um, So we were talking about prokaryotic eukaryotic cells, and we have this. We went through this web page to understand the cell structures, and now we are here. Okay, we saw that cell wall, and there is one more thing below the cell wall. That is plasma membrane. This is the plasma membrane. I'm going to discuss a bit in detail. This is how the bacterial cells look like, if it has a capsule like, and remember this bacteria is called Escherichia coli, E. coli. This is the generic name or genus name. Often we abbreviate it and we keep a capital E letter and dot. This is the bacteria E. coli, or you can full name, genus name Escherichia, species name coli. Why coli? Because it is found in the colon, in our intestine. These bacteria are found. As I said, these bacteria are normally they do not infect, non-virulent in nature. In our body, they are still present. There are some strains of this E. coli bacteria which can cause disease. Okay, it can cause dysentery. Often it can infect your internal organs. It can infect your lungs. Okay, so it's not that all of the strain which is still residing with you in your stomach, it is harmless, but the other strain, it is harmful. Can we completely do away with the bacteria and live happily? No, we cannot. We need certain number of microbes for our body to survive. You cannot kill all bacteria and expect yourself to be healthy, living healthily. So inside your gut, on your skin, there in these places you see maximum number of microbes present. Okay, so how do they look? They look like capsules, you know, see. And this is how. And do they have flagella? No. Can you see pili present? See, this is a scanning electron micrograph. With scanning electron micrograph, it is a bit of difficult to observe pili. Pili, or in singular you can say pilus, Pillus, pilus or pilus, whatever it is. Okay, so again, relabeling of it. This whole red color is the cytoplasm, because that's the actual fluidic area where things move. Then nucleoid. Then we have dots, like these are the only organelles. So nucleoid, it's not a proper organelle, but yes, it has this nuclear material. Then ribosomes. Then we have mesosomes, and finally we have this membrane. Okay, membrane and cell wall, which prevents. Then we can say organelle as flagella, which helps in motility, right? This is the function, you can see. It's the function, what it does. So what are the functions? Cell wall protects the cells. Of course, it holds the liquid cytoplasm together, and it's kind of a polythene bag which keeps which holds liquid curry when you order food from Swiggy or Zomato or something. Okay, it's the polythene bag sort of thing. But this polythene bag is nothing, doesn't have a complete imperviousness. This polythene bag is porous in this case. In case of bacteria, it allows things to pass in and out of the systems. Okay, but overall structure is given by the cell wall. Then we have a plasma membrane. This is a membrane, you know, if you do not have a membrane, you do not have a living organism. Okay, you have this liquid or gravy-like material, that is your cytoplasm. It has to be outlined by a plasma membrane, which is very flexible. Plasma membrane, you can imagine it as a water balloon. Water is your cytoplasm and balloon is the thin material. Now this water balloon is not so tough. It can give you any shape. If you just it do not have to be very rigid on that. If you throw it down, then it will burst, of course. But it holds the water together, and it allows transports or molecular transports of many things, including water, of course. Okay, this is so we call them selectively permeable membrane, meaning it doesn't permit everything, but something at some duration of time it can permit. Like it has this check post, and then this extension of plasma membranes through the cell walls, it comes out, and this is pili. So then we have cytoplasm. Cytoplasm contains whole, as we see, there isn't much of organelles inside the bacterial cell. Then still the bacterial cell grow, they divide, and they do binary fission, of course. This is the way they divide. And they do their own metabolism. So we understood that metabolisms is a set of a lot of reactions. Okay, that is one of the characteristic of life, right? Lot of reactions, catabolic and anabolic reactions happening together in a very homeostatic way, right? So for those reactions in living systems, this is our trick to be alive. We have got enzymes for doing these reactions. Okay, what are these enzymes? Enzymes are nothing, they are proteins only. Okay, but there's enzymes can give a specific groove or site. See, as if I tell you, any reaction, any organic reaction, if you see, there is this. So we will read detail about it, but enzymes are molecular catalysts, I can say. Any reactant 1, reactant 2, giving to product. This can happen faster if there is a catalyst in between, right? What does catalyst do? Catalyst actually brings this R1. Actually, if you really dive deeper into this reaction where the reaction is happening, so any catalyst, if it can promote the reaction happening, it's just that because it can absorb these R1 and R2, R1 and R2, hold it in a place closer enough for the product formation. Okay, so if this is possible, these catalysts we have learned in our organic chemistry in your 10+2 probably are palladium, platinum. We have sometimes we have microcatalysts like zeolite. Those can also work as catalysts. In living systems, we cannot afford to have palladium, platinum. These are very unreactive metals. So what we do is we make our own catalyst out of molecules what we can make. That is protein we can make, and we made a protein. We ask them to make an enzyme. That is a catalyst. Then we have the nucleoid. Nucleoid is actually, this is a scanning electron, transmission electron microscopy picture. With the light microscopy, you cannot see this detail of a prokaryotic cell. Okay, so transmission electron microscopy can that, yes, it also reveals a black spot here, but actually if you in a better picture, we can see a spiral arrangement here, and this is an this mesosome. And where is the nucleate? Nucleate is here. Why it is lighter in color? It should be dense, like nucleus is dense, because see, the cytoplasm contains maximum protein and ribosomes, which can give density to the, or radio opacity, or X-ray opacity, bit of like X-ray opacity. Sorry, electron beam opacity to this cytoplasm. That's why cytoplasm looks, although it is more liquid in nature, fluidic in nature, but it has got high electron catching molecules. Actually, there are more number of varieties of molecules are present here. Only in nucleoid, we have less number of varieties. Only DNA is present. Okay, this is how it looks through electron microscope. People have actually seen through electron microscope, and then they made some artist impression. This is how we. It is easy for us to draw this way. Okay, so how does it look? It doesn't have a boundary, doesn't have a membrane boundary, and the overall DNA is floating simply like that. So we can call it as a nucleoid. Well, by the time the nucleoid is being discovered, DNA wasn't a very common term. Okay. Then we have this ribosomes. Ribosomes are very small, double unit. Next class, I will show you how ribosome work while we will be discussing eukaryotic cells. Okay, this is ribosomes, larger unit, smaller unit, and it's very dense particles, and they actually assemble with a default. It doesn't need anything to assemble themselves. Once they are prepared, they assemble by themselves in a exactly right manner. This is the protein synthesis machinery where the DNA, this we will learn in the central dogma. But at least you know that it's a very important machinery which is used in eukaryotes, prokaryotes. It is used for DNA, protein synthesis. Then we have flagella. Flagella is often a whiplash system. If this is a membrane, flagella base is would be like this, and uh, so here the cell's job is to spin it up. Okay, it will simply spin it off, so that this whiplash flagella will keep on moving. So what does it do? It actually propels the, see, otherwise the bacteria is, you have seen it's like a capsule, or you can consider it as a worm. If it doesn't have a flagella, it has to wriggle. If you remember, wriggle. So it has to take wriggling motion. If it does, have flagella, if some, it's not all bacterial species have flagella, some do have, so it can really swim very fast. It's just uh, so here it's a what do you say, caterpillar wriggle, and here you can see it's almost a centipede or millipede, which can move very fast, a lot of legs. So fine, this is what the prokaryotic cells look like. But how about the membrane? This is the main cause. If you, in fact, all antibiotics, we actually take antibiotic to kill bacteria, right? So majorly all antibiotics work. I'm not saying all, major would be better. All major antibiotics target the membrane of the bacteria and and allow and drill holes into this membrane. That is all its job. So once they have hole in the membrane, then the selective permeability of the plasma membrane is lost. And if it is lost, then overall the life of a bacteria is also lost. Okay, so how does this membrane look like? Often in gram-negative bacteria, but this gram-negative positive, I'll just tell. This is the membrane. How does it look? You see, this is two membrane layers, and in between there is a gluey cell wall-like layer. And each membrane is constituted of phospholipid dilayer. This is a phosphate group. This is a lipid chain. Okay, phospholipid dilayer. So this is negatively charged. This is hydrophobic, oil soluble. Okay. So what is the thickness of the membrane? Often the membrane is 4 nanometer thick. So thin. Then you can see another layer above the membranes and cell walls. There is a slime layer. Okay, often we call it capsule layer. So capsid or capsule layer we call it. This is a slime layer. So I was just saying that can we see multicellular prokaryotes? This is the only case when we can see multicellular prokaryotes where there is no proper cell-to-cell communications, not very regularly, I would say. There is transient pili and the job is done. One nuclear material transported, job is done. But there is a extracellular matrix that's called the biofilm, which is often an extension of capsule. This is slime or gluey material produced by bacteria. I think yesterday we saw the under the microscope, we saw maximum bacteria were like this. They had a capsule and they were sitting inside, right? Okay, so we saw what are the layers we can have. So outer layer we can have a capsule, then we have cell wall, plasma membrane, then cytoplasm, of course, right? This is how the prokaryotic cells look like. Often these prokaryotic cells, they're below the capsule. They can be of two types. Now, I just told you that there is a particular stain called crystal violet, and a scientist called Graham, his surname was Graham, and he designed a particular experiment with which he can actually distinguish many species of bacteria. And with that, we started calling it gram positive or gram negative bacteria. Okay, the E. coli we just saw, that's a gram negative bacteria. Okay, um, and the lactobacillus you see in your which makes your curd, that bacteria is a gram positive one. Okay, so E. coli will look like this, and lactobacillus will look like this. Okay, same rod like, but lactobacillus is actually elephant, and your E. coli, you can say it's a dog size bacteria if you compare it shape size wise. So, so what is the difference? Why this crystal violet? Generally, the bacteria will stain pink. There is a pink color dye, but if the gram positive bacteria will additionally stain blue, so finally it will appear blue because of some crystal violet stain. Why it appears blue? Because of this additional thick layer of peptidoglycan, which can end up in teichoic acid and all. This is the additional layer. So these are gram positive bacteria. It's a very thick cell wall they do have. It's a peptidoglycan layer, which was very thin in gram negative bacteria, which is in between two membranes, inner membrane and outer membrane. Both are phospholipids. Okay. Okay, that means in a gram positive bacteria, you do not see outer membrane at all. You see the inner membrane, and the peptidoglycan layer is blown out. It's very thick. Okay, just to prevent antibiotics and other toxic thing to enter into its body. That's the only reason. Okay, then uh, and of course after this it doesn't have any outer layer membrane. Okay, so what is the nature of the chemical composition of the membrane? This we'll discuss later, so do not have to write. You can only write that there is a thick cell wall outside the cell membrane in case of gram positive bacteria, and in case of gram negative bacteria, here the cell wall is there, but it is very thin and it is sandwiched between two membranes, inner membrane and outer membrane, so that the cell wall isn't exposed to the blue color dye. If it is exposed also, it will be faintly blue colored, not very dark blue color. Okay. So that would be for the prokaryotic cells. Um, so what we can think now, I can just go back to the web page, and I can ask you to guide me for constructing the bacterial cells again. Animal cell, plant cells, we are going to study, don't worry. Let's construct a bacterial cell. We just studied that. So what are the parts we need? Can you tell me? You can say from the left and from the right, which portion should I pick? This is only the cell wall outer layer it has given. The only cell wall is given. To make the living bacterial cell, we need other parts also. What should we put? Should I put mitochondria inside? Should I put a mesosome inside? Yes or no? Yes. Should I put mesosome? I hope everybody is awake.
Sir, we cannot see.
Yes, sir.
Yes, sir.
So the
Okay.
screen is not.
Screen is not visible.
Okay. Fine.
No, sir.
Okay. What I did is I skipped the introduction. I just went to the last tab, that is construct a cell. So animal cell, plant cell, I am skipping. I am going for a prokaryotic cells. Okay. So here we have a lot of options, the more than the options what we have seen here. So we need to carefully choose the thing which exactly we need it in the cell. Here, the only thing which is provided is the blue line, that is the cell wall. Understood? So can we take for a prokaryotic cell, that is a bacterial cell, can we take mesosome inside? Yes or no?
Yes, sir.
Yes,
Yes, sir.
Yes, sir.
Let's take mesosome. Yes, it is fitting here. Can we take flagella?
Yes, sir. Yes, scouts.
Yes,
Okay,
sir.
flagella got fit,
Yes, sir.
not a problem. Can we take mitochondria? No,
No.
we
No,
didn't
sir.
see that. Can we take lysosome, peroxisome? We didn't see that.
No, sir.
No, sir.
Even if we click, sorry, it isn't a part of prokaryotes. Can we take a nucleoid here?
Yes.
Yes, sir.
Prokaryotes
Yes, sir.
do have a nucleoid, doesn't have a proper nucleus. So if I simply click at nucleus, I see that it's not a part of prokaryotic cells. It's C, right? Can we take chloroplast?
No, sir.
No, sir.
But
No, sir.
but we know some bacteria are photosynthetic. Isn't so?
Sir, it is a part of plant cell, no sir?
Yes, it is a part of plant cell. Some bacteria are photosynthetic, but they do have their photosynthetic machinery inside the cell. Doesn't have a specific organelle for it. Okay. Now we have another organelles called endoplasmic reticulum. These are membrane-bound organelles present close to nucleus in a eukaryotic cells. Do we have? No, we cannot have it in
No, sir.
prokaryote. Can we
No,
have
sir.
vacuoles? No. Can we have pili, small thread-like
Yes, sir.
structures on
Yes.
the?
Yes.
Yes, sir.
Yes, pili is fitting here. Can we have ribosomes? Very small,
Yes,
small
sir.
Yes, sir.
particles spread inside the cells. Yes, ribosomes are there. Good, good. Then can we have Golgi
No,
apparatus?
sir. We don't take.
No, sir.
No sir.
So
No,
that
sir.
means what do we
No, sir.
discard out of it? These are all actually organelles. What do we do not have is any membrane-bound organelles. We do not have it inside the cell. Okay, these all are membrane-bound organelles. Even nucleus is membrane-bound in eukaryotes. Chloroplast is membrane-bound. Membrane-bound is meaning an organelle which is made up of membrane. Then we have this endoplasmic reticulum. These all are membrane bound, and we do not have space for that. In fact, many bacteria are of a size of an endoplasmic reticulum or the overall size of a Golgi apparatus. So you cannot have this thing which is of the same size inside a bacteria, right? So we understood that these are the basic things in the bacteria. With these basic things, a living system can function. Well, what does eukaryotes have? They have a luxury thing. Okay, they have a lot of features, and these features are also not useless. These features for eukaryotes, we need that for multiplications or coordinations among multiple cells. Bacterial cells, they are not true multicellular organisms. Okay, often you can find them in lumps or patches, but they are not multicellular in nature. Okay, so that would be all for the class today. Thank you very much.
Thank you, sir.
Bye. Take
Thank
care.
you, sir.
If you have any
Thank you,
doubts,
sir.
you can write the comments in the class page itself, so that I'll be happy to answer. Thank you very much.